All right. So basically, today I'm going to talk about sort of the basics, as I mentioned to you, kind of the cast of characters of cells and organs of the immune system. The slides are posted. Yes, I will. It's more slides than I could get through one day. I will have to do some of them on Friday. I got overly excited. I had way too much fun putting this together. So that's that. For reasons that I do not understand, my software that lets me do screen recordings to make the YouTube videos is being weird. So I probably won't get videos up until like at least this weekend because I'm going to have to do some troubleshooting. It worked in my office like 2 minutes ago though. I don't know why it doesn't work now. That's okay. Tomorrow in lab, I'll have sort of the lab syllabus and schedule for you, as well as a handout of the activity that we're doing. We're using prepared slides and doing microscopy, so it's one where like lab coats, things like that are less important because it's prepared slides. And it is related to cells of the immune system, so that's kind of why I like to do it right now. Like I said, we'll have a week off of lab sometime in the middle of the semester instead of having one off at the beginning of the semester because that's when we're more stressed. So that's when the free time matters more. All right. Oh, and as I mentioned, I had way too much fun putting some of this together. I forgot to mention this on Monday. Some of you guys probably already know this. I do a lot of science communication and podcasting. I am co-host of an immunology podcast called Immune. There are a bunch of things that I learned from the podcast that like I updated from last year because of stuff I learned on the podcast. So that's why I was having so much fun. So when I reference it today and throughout the semester, that's what it is. You're welcome to listen. You don't have to, but it is a pretty great podcast. So I left off giving you guys a little bit of information about microbes because the immune system, as I told you, really evolved in response to the threat of pathogenic microbes. And so we need to remember a few key things about microbes if we're going to think about immune defense against them. A couple of these I'm going to flip to other slides because I actually have a whole slide on them, but this is the overall list of things to think about in terms of microbes. So one of the biggest things, and this will come up today, and this will actually be coming up relatively early in the semester, has to do with replication rate. My example that I'm going to use for you guys is an example of bacteria. That, to be honest, it would be even worse if I used viruses. So this is something that I talk about in microbiology, which is sort of a standard growth curve for bacteria, like E. coli or something. You don't need to know all the details of the standard growth curve, but what I want you to notice is that at the beginning of this growth curve, we start out at like 10 to the 5th, which is 100,000 bacteria. And in, 5 hours, we're up to something like 10 to the 8th or 100 million. And so you can, in general, E. coli, for example, will double in about 20 minutes. You'll go from 1 cell to 2 cells in about 20 minutes. Your cells, the fastest that one of your cells can go through cell cycle, this is an asterisk because I already know a paper that disagrees with what I'm going to say, but we're just going to go with the textbook answer, not this paper. The fastest your cells will go into cell cycle is once every 24 hours. So if you had to wait for one of your cells to divide to deal with some of these bacteria, you would have basically, the bacteria would have won. You would have gone from 10 to the 5th or 100,000 up to like good 10 to the 10th, so 10 billion bacterial cells. That bacteria is going to out-replicate you every time. And so speed of responses and sort of dealing with just what I like to think about is like just knocking down numbers so that your cells have some time to catch up becomes really important. And so this is just, I'll say more about this, as we, go forward, but this is a phase of, immune responses that people often kind of think about as a very classic phase of the immune response. I'll say more about it, and notice it doesn't even start really doing anything in this particular phase of the response until day 7. If this was really the only immune response and you had to wait till day 7 before you did anything about this bacteria, you'd die every time because the bacteria would out-replicate you every time. And so thinking about replication rate, thinking about, how we're going to knock those numbers down is going to be really key. And that's something that we're going to be seeing a bunch. We want to think about how microbes may or may not be different from our cells, and do they offer unique targets? So bacteria are prokaryotes. Their cells are pretty different than ours. Fungi and parasites are eukaryotes. Their cells can be much more similar than ours. So it's harder to have a different biochemical target that distinguishes against those cells. Viruses mostly use our cells' machinery. So again, it might be hard for them to have unique targets. And so that's one thing that we're going to have to think about when we think about how we deal with some of these microbes. We want to think about mutation rate. How might we have an immune response that deals with a microbe that's changing all the time, especially when we're not going through generations nearly as quickly as it is. We want to think a little bit about where microbes live. So we're going to have to think about immune responses that act against pathogens that are outside of cells. We're going to have to think about immune responses that act against microbes that are in the cytoplasm, and sort of the classic microbe that's in the cytoplasm is a virus, or microbes that might be in different cellular compartments. And so the way you defend against each of those things is going to be different. We also are going to want to think a little bit about how big microbes are. So here you can see, viruses 10 to the -7th meters. They're pretty small compared to our cells. Intracellular bacteria, some of the extracellular bacteria, still relatively small compared to our cells. Even fungal cells are a little smaller than our cells. Parasites can be sort of closer to on the order. I showed you guys this last time. This is Ascaris, which is a parasitic worm. This has come out of someone's GI tract, and it is in someone's hands. So that gives you scale. What I normally will say here is that if you have something that big, and you think of what people often think about as immune responses as they think about things like phagocytosis, you weren't phagocytosing that. Look how big that is. How the heck is one of your cells going to eat it when it's that ginormous? So clearly you got to have a few different options here for defense. I will mention that I did a podcast episode last week where we were talking about some stuff getting phagocytosed that I would have originally told you was too big to be phagocytosed. So maybe my number's off, but I'm still pretty sure you can't phagocytose that. So I showed you this as kind of my thought about the immune system and sort of all the complexity and all the cells and all the interactions that we have in the immune system. And I told you last time that we have a few ways that we like to divide up immune cells. And so I want to show you some ways that we as immunologists like to divide up all of this mess and try to sort of organize it. The first way that we will talk about organizing immune responses is into a set of layers. I've got 2 different versions of this from 2 different textbooks to kind of help us think about this. And so there are different kind of parts of the immune system. There are some things that are known as barrier defenses. There's a part of the immune system known as the innate immune response, and there's a part of the immune response known as the adaptive immune response. If you may have previously heard in other courses about something called acquired immunity. If you've heard of acquired immunity, that is the old-fashioned term for adaptive. So acquired, we don't use anymore. We stopped using it like when I was very much in the beginning of grad school. Like, to my knowledge, we stopped using it more than 20 years ago, but it still shows up in some textbooks and makes me a little nuts. So if you see it, it's adaptive because you didn't acquire it from anywhere. You had it all along. It just got better and adapted. And so we've got sort of these three different aspects of immunity. I'm going to say a little bit about barrier defenses today. And then we're actually going to come back and there's going to be a day about barrier immunity later on in the course. So that's that. Innate immunity will be spending the next sort of section of the course. So starting on Friday and then like the week after and a little bit of that, we're going to be talking about innate immunity. And for a bunch of time in lab, we're going to be thinking about innate immunity as well. And then we also have adaptive immunity. I also really like this image from my microbiology textbook that's kind of trying to get at the same thing, where it's showing sort of this defense of this castle and the fact that there are multiple different types of defense that sort of overlap and work together. You know, we've got a moat, we've got alligators, we've got archers, we've got arrows, we've got boiling oil, all trying to attack against the invaders. And so the thing that is important to note about this, and the reason why I show this, is that sometimes people think about sort of these layers, the layers this way, as like you have to deal with one versus the other, or that they're totally separate and don't work together. But in reality, it is a bit more like this. In sort of many of these things may be ongoing at the same time and may be kind of interacting and playing off each other. They're all different, but they're all doing something unique. They're all important, even if sometimes people underestimate the importance of some of them. So with barrier defenses are really specialized types of cells and organs that try to keep microbes out of your body. So the biggest barrier immune organ is your skin. And the idea here is kind of that it has to do with that, like, keep the numbers down, keep the microbes out as much as possible. And so we have skin that's going to keep out microorganisms. We also have other kinds of barrier defenses in parts of our body that are in association with the outside world. So a particularly famous thing is that your stomach is super acidic. So if something happens to get into your body and gets into your stomach, the acids might kill it in the stomach. We also have things like in your respiratory tract, we have cilia that are kind of flushing things out. We've got digestive enzymes in a number of places. We have, again, you can see cilia sweeping outwards. You've got coughing. You've got the fact that you're actually flushing the urogenital tract with urine to flush things out. And so we actually have, tears, that may come out of our eyes that are going to try to flush things out. And so all of those are sort of physical barriers that are going to block colonization. And so in most cases, these are really stopping infection, not so much stopping disease. Most people, I'm not going to, again, like I said, say much more about barrier defenses until later in the semester, and most people forget about barrier defenses. However, if you were to work with a burn victim who has lost their skin barrier in the hospital, the main issue they have is many, many, many bacterial infections because people just underestimate how much this is actually a thing. One other thing that I will mention here is that there are a couple places where this image talking about some of the specialized tissues and the barrier defenses mentions in a few places some antimicrobial peptides or some other things. And some of those I personally think of as being part of the innate immune response. And so even though I can show you this image of, oh, these are the three things. This is the barrier box, and this is the innate box, and this is the adaptive box. The immune system doesn't know it's supposed to be in boxes. And so there are situations where some of these things kind of blur lines a little bit. One of the other kind of, just sort of to give you a general idea with a lot of our barrier organs, things like the types of junctions between cells are really important, and other aspects of sort of the anatomy of the actual organ, because you don't want to have microbes be able to slip in between the cells. And so you can see it is actually a physical barrier. There are some chemical mediators that are made, though, again, I'm not going to focus a ton on those. Those will come up more in a later time. And in fact, the microbes that normally live in those locations are an important part of the barrier as well, because they fill the ecosystem. They fill the niche space so other organisms cannot colonize. If you've ever heard that when you go on antibiotics, you're supposed to eat a lot of yogurt, it's because you're killing those commensals in your GI tract, and you want to make sure that you're filling the niche space with good bacteria and not letting it stay open for bad bacteria. So in fact, the barriers, you can kind of think of like, those aren't even my species, and yet they're kind of part of my barrier organ. That's weird. So that's kind of the big picture for barriers right now. So then the other layers that I want to talk about are the innate and adaptive layers. And a key part of thinking about this is that I want to kind of give you, like, really at this point, kind of some definitions of what is the innate immune response and what is the adaptive immune response. We are going to spend whole sections on innate versus adaptive immunity as we go forward. So it's not like you need to know every detail of either of them now, but as I start to talk about some of the cells later today, I'm going to say, like, this cell is part of the innate immune response. And so you probably should know what the innate immune response is before I tell you that which cells are part of which response. What I will also say is that adaptive immune responses are often the sort of stars of the show, or the ones people think a lot about. So on Monday, some of you guys, we were talking about defining the immune system, and some of you guys name checked some specific cell types. Every cell type that got name checked was an adaptive cell. And so, yeah, that's part of the immune response, but there's this whole other part of the innate response. Adaptive gets a lot of the press, but we should think a lot more about both types. So there are there are a few different things that are sort of part of the definition of innate versus adaptive immunity. So one of the big differences between innate and adaptive immunity is timing. With the innate response, we are seeing a response within hours. And so usually the innate immune response is particularly important within the first 96 hours after infection. While with the adaptive immune response, we're talking days to weeks. Remember how I showed you that sort of hypothetical thing, and I was like, look, you had to wait 7 days. That was an adaptive immune response. So fortunately, you don't actually have to wait 7 days, because you also have this other part of the immune system doing something. And realize that that's kind of important with that replication numbers problem I told you. You need to have something fast if you're going to keep those microbial numbers low. And you can't necessarily just wait around for the adaptive response. The next 2 points on here are super important points for immunologists, and are things that are really hard for me to explain until I explain them in super detail. So I'm going to sort of big pictures them, and you might be like, I want more on that. Don't worry, you are going to get so much more later. But the basic idea is that with the innate immune system, there's a relatively small number of receptors, while the adaptive immune system has a crazy huge number of receptors. And we get that crazy huge number of receptors based on some unique genetic processes. And so that's kind of one part of what they're trying to get at here with fixed versus variable and limited specificities versus highly specific. The other thing that they're trying to get at here, and again, we'll see much more of this, is with the innate immune system. The innate immune system is kind of recognizing broad features of a microbe. So it might recognize, hey, look, it's a gram-negative microbe. It isn't going to say, oh my gosh, it's E. coli, or oh my gosh, it's Proteus, or oh my gosh, it's Salmonella. It's going to be like, gram-negative microbe. Whereas the adaptive immune system is going to recognize, oh my gosh, this is COVID variant, or SARS-CoV-2 variant KN221B. It's going to be super specific to, this year's flu versus last year's flu versus this kind of E. coli versus that kind of E. coli. Whereas, innate immunity is going to be like, it's an RNA virus. It might be SARS-CoV-2. It might be influenza. Who knows? It's just an RNA virus. So there's also kind of this broad thing with that smaller number of receptors versus this crazy specificity. Like I mentioned, the specificity in the adaptive immune response comes from some unique genetics processes that we'll talk about a bit later. And in a lot of ways, the ability to do that genetics process is sort of what we're getting at here. So when we see that process, that will make hopefully some more sense. The other big difference is sort of how the response looks over time. So the innate immune response is basically constant over time, while the adaptive immune response gets better over time. It adapts. It shows a property of memory. And so you can see all of this depicted here as well. So you can see the innate immune response to a particular microbe in this lighter blue and the adaptive response in the darker blue. So notice the innate immune response is doing something pretty early, while the adaptive response is taking a while. So you can see that timing difference. But you can also see that if you were infected with the same thing a second time, the innate response looks the same every time, and the adaptive response looks way different. It actually changes and gets better both in terms of size, in terms of strength, all of those things. And so you can see this is sort of the other big defining feature of adaptive immunity. Because there's sort of this unique genetic process that I mentioned, and there's this ability to have memory and to adapt to a response, people have spent a lot of time studying adaptive immunity. Like I said, adaptive immunity gets a lot of the press. But I do want to just point out one quick thing about innate immunity. I will tell you that when I was a grad student, and we were TAing immunology, we always joked about like, oh, who has to talk about innate immunity, the boring stuff. Nobody wants to talk about that. Since that time, there have been multiple Nobel Prizes given for discoveries in innate immunity. Innate immunity is by far the hottest area of immunology. It's what my lab works on and many other work on. So it's really pretty cool. But if nothing else, I give this slide to grad school me to be like, hey, you, innate immunity is really important. Don't underestimate it. And so here you can see number of pathogens on the y-axis and time on the x-axis. And in the yellow graph, you see kind of a normal human. And what you will notice is that normally you get infected with a pathogen, that pathogen reproduces for a while, and eventually the number kind of stops increasing. And eventually we get rid of that microbe and you feel better and you live happily ever after. There are people who have defects in their adaptive immune system. So these are immunodeficiencies. These are diseases where there is too little immune response. In those individuals, you can see that the microbes increase, and maybe they increase a little bit more than they would have otherwise. But the big thing is that they never actually get cleared. They kind of get to the max number and just stay there forever and ever and ever. And this person is, these people tend to have pretty severe disease. Most of them sort of don't make it to age 20. Well, before we had treatment. Now we have treatment, so they're all good. But in red, you can see theoretically what would happen for a person who didn't have an innate immune system. And what you can see is that the microbe numbers increase even faster and basically never even get controlled. They just go sort of to infinity. This is actually theoretical. We have never actually found a person who completely lacks an innate immune response. It seems as though that is not compatible with being alive. So you can live without an adaptive immune system. You can't live without an innate immune system. So while the adaptive immune system is going to be the thing we spend most of the semester on, is going to be the thing most people think about when they think about immunology, don't sleep on the innate immune system. So I showed you this quote on Monday, thinking about what the immune system is. And I'm showing it to you again because what you'll notice is that when Mark Davis describes immune responses, he kind of talks about cells, he talks about molecules, he talks about organs and a circulatory system. And that's really how a lot of other ways we divide up the immune system. And so today I'm going to really be spending time talking about who are the cells, as he mentions, and depending on how far we get time-wise, we'll be talking about sort of the organs part. If you look at the course schedule, you'll also notice that on Friday I use this word called humoral. I say that on Friday we're going to talk about the humoral innate immune response. That actually means, realistically, when an immunologist means humoral, they mean that the molecules are important more so than the cells. And so that's another distinction that sometimes immunologists make in terms of, are we talking about molecules, are we talking about cells? I don't love that distinction myself, because I remember being an undergraduate in immunology when I was hearing about humoral immunity and being like, but the cells make the molecules, so why is this not cells? So I don't love it, because it's not like the molecules come from nowhere. They come from cells. But there are some reasons why that distinction, especially when we talk about innate responses, is going to be really important. So one other thing I will tell you is that I've had this sort of classic way of discussing all of this stuff for a long time. And the reason why I had so much fun putting all this together is that in the past year, this paper was published, that we talked about on the podcast, which is why I was getting so excited, where someone actually did the math about all sorts of different things about immune cells in a reference 73 kilogram man. They did so much math. And so there are a couple of points that I learned when we were reading this paper that have tweaked how I normally talk about this stuff. And so I'll just tell you that that's one thing to point out. And I'm actually going to start with one key piece of information that I want to mention that did come from that paper. And that really has to do with location of immune cells. If right now I was going to measure your immune system, if I was going to study some of your immune cells, what kind of sample would I probably take? Yes, David.
Blood sample?
Probably a blood sample. Immunology actually has a pretty old history because immunologists have been able to get blood samples for a really long time. Neuroscientists have not been able to get human brain cells to play with for very long, but we've been able to get human blood to do experiments on since the 1700s. So oftentimes we think about immune cells and cells of the blood. And we are going to talk a lot about cells of the blood. Immunologists do spend a lot of time thinking about cells of the blood. What I want you to notice here they actually were looking at this reference man and looking at where all the immune cells were. And notice blood, how many immune cells were in blood compared to some of these other locations. And so what I want you to notice is that, honestly, for most of this semester, we're going to think about blood. We are going to think about lymphatic system. We're not going to always talk about some of these other locations. And so just know that one of the big things in immunology is wait a minute, are we missing things because we're not looking at people's immune cells in some of these other places. And so sometimes I might mention today when I talk about different cells, this is a cell that spends its time in the blood, or this is a cell that spends its time in the tissue. And so tissue immunology is sort of a really hot area of immunology now in terms of not just thinking about straight what's going on in the blood. And so you can see sort of this reference. The different colors are actually different types of cells. So you can see that the different parts of the body actually will have different representation of different types of cells. I also show you this image just to show they were actually looking at number of cells per mass of organs, and you can find immune cells. You know, there's some that have a lot, like tonsils and spleen and thymus and lymph nodes, but like if you can name a tissue, you can find immune cells there. And so we are mostly going to be focusing on immune cells in sort of the blood and lymphatic system, but that may in the field not always be giving us a total picture. I'm going to tell you a bunch about some of these different cells going forward. But I just wanted to tell you one other thing that I learned from that paper. I'm going to be telling you about some of these different cell types on the next few slides. One of the things I actually truly did not know until I read this paper, until we discussed this paper on the podcast, is some of these cells actually vary not only in number, and that's what I'm going to show you a lot on later slides in terms of like some of them are really common, some of them are really rare. They actually vary a ton in mass too. Like, and we're going to look at them under a microscope tomorrow. Like I know like the approximate sizes. I work on a cell called a macrophage, which is this. Macrophages are like heavy, like you would not believe. If you look at the number of immune cells, macrophages make up this small fraction. If you look at the weight of your immune cells, macrophages make up 50% of the weight of your immune system. And so and note that I learned this, and many immunologists learned this in 2023. So while I'm going to tell you about, things that are unique and different about some of these different cell types, know that there's probably still a lot to learn about all of this. Usually when we try to do our distinction of different types of cells of the immune system, we focus on the blood. And so here I'm showing you kind of how we divide up cells of the blood. And we are going to be seeing prepared slides of blood tomorrow in lab. And so these are the cell types that can be found in the blood. One big cell type that's found in the blood, perhaps unsurprisingly, is a red blood cell, which hopefully you are aware of the fact that blood is red. And so those red blood cells are typically known to immunologists as erythrocytes. I'm only going to say tiny bits about erythrocytes throughout this semester. They have one really important distinguishing feature, which I guarantee you will know by the end of lab tomorrow, because it will be so obvious under the microscope that I'm not even going to tell you what it is, because you're going to look and you're like, oh, duh. There also are some structures called platelets that help in clotting. Platelets actually come from another cell type called a megakaryocyte. And then the cells that we generally are spending our time thinking about as immunologists are the white blood cells. Another word for white blood cell is leukocyte. And so we're really going to be thinking about leukocytes. Most of the time tomorrow, you're going to be spending time thinking about leukocytes. And we can divide up those leukocytes or white blood cells. When we divide them up, we tend to divide them up into three big groups. Those groups are the granulocytes, the monocytes, and the lymphocytes. Again, I'm going to say so much more about all of these, and by the end of lab tomorrow, you will know these cells. One place where students sometimes will get confused about this is that sometimes people have trouble with the words lymphocytes and leukocytes. Lymphocytes are a type of leukocyte, But leukocytes is like the whole big group of all white blood cells. Lymphocytes is just like a small subset of them. And so those two words are not interchangeable. When we think about all of these cell types, one of the ways that these cell types vary is in terms of their frequency. But note, this is frequency in the blood. So sometimes we look at some of these cells and I'm going to be like, oh, that cell is super rare. But that's because we usually count it in the blood. And maybe if we actually counted it in the whole body, it wouldn't be so rare. So note that we're a little tricky here. And so you can see for red blood cells, we have about 5 million per cubic millimeter. While some of these other cell types were more like in the hundreds per cubic millimeter. So big differences in frequency between all of the types of cells that we're talking about. If we just think about the leukocytes, you can see big differences in what percent of the white blood cells are each of these different types of leukocytes. And if you've gone to the doctor and had a complete blood count or a CBC, you're basically getting this percentage figured out. One thing that's always pretty weird is notice how big the ranges are in some of these cases. Like, healthy range for humans is 50 to 70 neutrophils. Like, that's a ridiculously large range. And in a lot of ways that we probably should do better. And you can see some of these also have similar ranges. As I mentioned, I learned recently that there are big differences in mass in these cells, that I didn't totally realize before. There are also big differences in things like lifespan of these cells. So some of these cells you can see are listed as living for a certain number of days. I'm going to mention a few things about, we're going to hear about neutrophils a lot. Neutrophils you can see live between 6 hours and 2 days. So really short-lived cells. You are making new neutrophils all the time. Whereas lymphocytes, you can see, for example, are listed as living for days to years. There's a pretty famous paper. It was looking at certain type of mouse lymphocyte, and it showed that, and the only reason they came up with this number is because they stopped the study, that the lymphocytes could actually live 3 times the lifespan of a mouse. So 3 times longer than the mouse that they came out of. And that was just when they ended the experiment. The cells were still fine and could have kept going for a while. So 6 hours to 3 times your lifespan. So you can see there's a bunch of variation there between all of these cell types. But one of the things that is really important to think about with these cell types is that all of these cell types that I've mentioned come from the same original stem cell. So all of these cells that we're talking about all originate from this cell called the hematopoietic stem cell, or HSC. Hematopoiesis is the development of all of these blood cells, and that word is up at the top. That comes from 2 Greek roots. I know how to speak. Comes from 2 Greek roots, hema, blood, and poiesis is actually the same Greek root as poetry, and it means to create. And so it's creating blood. The number of times I have heard immunologists make jokes about the poetry of blood cells, I'm a little done with that joke, but that's okay. And so all of these different cell types, even the red blood cells that we're not going to spend much time talking about, come from this original stem cell, the hematopoietic stem cell. Those hematopoietic stem cells are found in your bone marrow, and so this is a big part of what's going on with bone marrow transplantation. And the idea here is that this stem cell will get different signals and will develop to make all of these different cell types that we're going to be seeing in the immune system. What you can also notice, if you look at this overview of hematopoiesis, is that they're kind of like little branches, like there's little groups. So you can see there's this little group of cells here. Here's this little group of cells. And those groupings that happen during development of the blood cells match this grouping that I gave you here perfectly. This grouping was something that people discovered by looking at cells under a microscope a long time ago, but it actually makes sense in terms of the way these cells develop in that grouping. So one branch I'm going to talk about is really this branch that's shown here. So I'm going to start with this sort of central branch. This is how your textbook shows us now. You were just looking at an image that was from a previous edition of your textbook. Your textbook now blows it up into like one branch versus the other branch in a slightly different way. So there's the same stem cell, and now we're just focusing on one branch and saying like, yeah, it could also go somewhere else. And this branch is a branch of cells that are known as the myeloid cells. Another sort of the key thing to know about the myeloid cells is that the myeloid cells are the cells of the innate immune system. And so all of these cells that are part of the innate immune system, also red blood cells that I'm going to say relatively little about, all come from this myeloid progenitor. And so your innate immune system is basically this one sort of branch of development of these cells. And you can see that there are sort of there are three different parts, the red blood cell part, that whatever. But then you can see there's sort of these two other parts, and those two other parts are some of the subsets I told you about before. One of those is this sort of middle group right here. Those cells are known as the granulocytes. Granulocytes are innate immune cells or myeloid cells, and they got named granulocytes because they have granules in them when you look at them under a microscope. Basically, you can see some nicely here and here, they have all these little dots in their cytoplasm. Those are all little vesicles of stuff that those cells could secrete to other cells. And so you can see the granulocytes here. One other thing that is particularly important about especially the top one, or the neutrophil, is you can also see the nucleus of the neutrophil, which is in the dark purple. And you can see it's not like a nice spherical pretty nucleus like you normally think of. The nucleus has these little lobes. And it can have a bunch of little lobes. So we've got three different types of granulocytes. We have the neutrophil, which you can see here, is shown kind of with a pinkish background, and the purple is nucleus. You've got the lobed nucleus. The granules are a little harder to see with the contrast, at least in this image. We also have one called the eosinophil, and we have one called the basophil. And they differ in terms of the colors, basically which stains pick up the granules, how they look compared to their nucleus, sort of what we got. The neutrophil largely is part of the innate immune system, acting by doing phagocytosis. And you can see eosinophils and basophils are both listed as largely being involved with parasites. So I may have made a joke on Twitter this morning. I actually have a full set of I have a macrophage necklace, which is one of the cells we're going to get to, and I have a full set of every cell type earrings. But when I was putting on my earrings this morning, I was missing one of my basophils. I'm short a basophil, and I was actually kind of excited about that because basophils are also involved in allergies. And I was like, maybe if I don't have a basophil earring, that means I won't have my allergies. But I don't think it's actually going to work that way. One of the big things to know about these three cell types, the neutrophil, the eosinophil, and the basophil, is that they do vary pretty dramatically in their frequency in the blood. So you can see that neutrophils are by far the most common leukocytes, while eosinophils and basophils are much more rare. In particular, basophils are super, super, super rare in the blood. I often think of there's no basophils in the body, but it might just be because they're in places other than the blood. So that's one thing to be aware of. We will spend lots more time on all of these cell types as we're talking about innate immune cells. But one thing I just wanted to tell you briefly about neutrophils. Neutrophils, like all of these cells, sort of develop in the bone marrow. If you have ever had your white blood cell count measured and been told, oh, you have a high white blood cell count, particularly if that means you must have an infection. What that usually means is that your bone marrow released a ton of neutrophils to go be the SWAT team. And so those neutrophils will leave the bone marrow, go into the blood, and then eventually go into the tissues to try to clean up those microbes. And so, a high white count usually just means you have so many neutrophils. One of the other things that's pretty cool about neutrophils, you can see here that the neutrophils come from the bone marrow into the blood, and then they've got to go into your skin or wherever you have this infection. And they actually have to squeeze through those cool specialized barrier cell layers. So that's a whole process we'll talk about later. That they actually go through this shove through the crowd squeezing process. That's why their nucleus isn't round. Because if their nucleus was round, it would get sheared every time they went through a cell layer. So they have to have a weird shaped nucleus so they can fit through a cell layer. Tomorrow you're going to be seeing a lot of neutrophils, and you're going to notice them by their nucleus. You're going to be neutrophil. So know that actually isn't just a fun microscope thing. It actually has an important biological purpose. There are a number of other types of granulocytes that we don't find in the blood. The most famous granulocyte that is found in tissues instead of blood are mast cells. And so you can see a mast cell here. Again, it's got a whole lot of granules. This is another view of a mast cell where you can, again, see a whole lot of granules. The idea is that mast cell will release all of those granules, basically shoot toxic bombs out of it. This is ideally something maybe you could do if you were trying to kill one of those great big parasites that you can't phagocytose. So you shoot toxic stuff at it. These tend to live in tissues, not so much in blood. These are also my allergy problem children. And are likely related to basophils, but we have relatively few basophils to study, so we can't actually totally figure out all those relationships. So that was one branch of these myeloid cells. Another branch of the myeloid cells is this branch that you can see over here to the far right. These cells I'm going to kind of broadly talk about as cells called monocytes. If I'm being very technical, we have a cell called a monocyte that lives in the blood, and if it goes to the tissue, it will differentiate a little bit to become a macrophage. I am largely, for our purposes, going to call monocytes and macrophages interchangeable. I'm going to use those two words interchangeably, though technically monocyte is the blood version and macrophage is the grown-up tissue version. And again, these are some of my faves because they're what I work on in the lab. And they largely work by phagocytosis and killing microorganisms as well. You can see that they're not as common as neutrophils, but they are more common than the other types of granulocytes. When we talk more about sort of how all of these cells are working next week, we'll have to think carefully about what is a neutrophil. Why are neutrophils and macrophages different from one another? Because right now I've told you they're both just innate immune cells that do phagocytosis, but they actually do have a lot of distinguishing details. This branch of immune cells also includes one other type of cell, and that cell is called a dendritic cell. Dendritic cells are also sort of in this monocyte macrophage lineage. They are not found in the blood generally. They're really found in tissues. And I mean, look at this. This is an actual image of a dendritic cell. If you had that flowing really fast through the blood, all these little arms would get torn off. That cannot go in the blood. I think the only cells that go in the blood are like nice and round and happy. That's not going to live in the blood. It's going to get torn up. And so these are really tissue cells. They probably also are coming from monocytes. And dendritic cells are tricky because a big part of what dendritic cells do, they are 100% innate immune cells, but they turn on the adaptive response. And so they're, again, one of these places where we're going to be very much bridging these two responses. I'm probably going to say the most about them when we're getting towards adaptive immunity, actually, because of their role in starting the adaptive immune response. So that was one branch of all of these immune cells. But there is also this other branch. So here's our myeloid precursor. And this would be the myeloid stuff we just talked about. Now it's not there anymore. Now we can't see it. And instead, we're going to look at the other sort of piece of this, which is development into a group of cells called lymphoid cells, which is just a fancy way of saying lymphocytes. It's the same thing as lymphocytes. And the lymphoid cells, or lymphocytes, are the cells of the adaptive immune system. You can see that they branch out into lots and lots of different cell types. There are moments where sometimes I look at my fellow immunologists and I'm like, guys, we don't need to have a special name for the kind of lymphocyte that lives in your left pinky to attack fungi on Tuesdays. Like sometimes there's some places where we're really dividing them up really fine. We're going to divide them up somewhat fine later in the semester. Right now, lymphocytes. Those lymphocytes, there are a bunch of different ones, but really, these are the cells that you might have heard of, the B cells and the T cells. So B cells and T cells, they are the two big types of lymphocytes. They are the cells of the adaptive immune system. They are the stars you've probably, like I said, heard of before. Don't worry, we're going to get so much about those B cells and T cells. And you can notice that those lymphocytes are not usually as plentiful as the neutrophils, but they're sort of the second most important or second biggest group in the blood. If you were to look at the syllabus and try to say like this many days of which cell type, lymphocytes would get like 80-something% of the syllabus. So note that I like to point that out because sometimes when people get to the end of the semester, they're like, the immune system is all about lymphocytes. And I'm like, yeah, but notice they're actually a relatively minor component compared to some of the others. So know that the amount of time we spend on these and the amount of cells is not going to match up perfectly. And as I mentioned before, the numbers actually look a little bit different if we try to look at whole body. Neutrophils still win, but it's not as dramatically different. And if we actually look at number versus mass, like I said, no idea. Macrophages are like, we called the episode Big Macs because we were all like, they're how heavy? In looking at this. So again, remember that everything I've really told you is sort of the really super classic version of a lot of this and largely came from studies of the blood. And so when I told you, for example, that there are very few basophils, that's really there are very few basophils in the blood. And if we were to we can't do this, take apart an entire human body and count how many basophils, including the ones that are between your skin cells here, we might actually find out there's a bunch of them. We just weren't measuring them before. But this also lets me segue a little bit into, as we think about the locations of these cells, to the organs of the immune system. So as we mentioned on Monday, there's not kind of like the one big organ like the brain in the nervous system or the heart in the cardiovascular system. There are a lot of anatomic locations that are involved in the immune system. Just like with the cells, when we think about the organs of the immune system, we divide them up into groups. And so we've got some groups of immune organs. Though this is actually doing a little bit of color coding in that the red and the yellow are some of those different groups. So the first group, which I'm going to tell you a little bit more about in a second, are groups that are known as the primary lymphoid organs. There are three groups. After we talk about the primary lymphoid organs, I'm going to start talking about the next group. I'm probably going to run out of time in the midst of the next group. But that next group is called the secondary lymphoid organs. What do you bet the third group is called? The tertiary lymphoid organs. You can see, immunologists, we're so clever. We're very creative here. Everyone looks at all these terms and like, oh, this seems so complicated. But no, it's primary, secondary, tertiary. Not that complicated. So the primary lymphoid organs are sort of, well, it's pretty straightforward to understand. There are two. They are the bone marrow and the thymus. So just those two for primary lymphoid organs. They have interesting structure that we can see under a microscope. We can think about all the different sort of places in those locations, which we can think about later. But the definition of a primary lymphoid organ is that primary lymphoid organs are where immune cells develop. So basically, this is where immune cells sort of go from being a stem cell to being a ready-to-go immune cell. Sometimes we talk about them as, particularly the thymus, as like the college of the immune system, where the cells are going to learn how to do their stuff. So these are the developmental organs. Note that I'm going to say, this happens during development or whatever. You are doing development of immune cells every day of your life. So you are making some of these new cells all the time. Bone marrow is spongy tissue inside of bone. The thymus sits directly on top of the heart. It is not the thyroid, which is an endocrine organ that's up in the neck. People sometimes get the thyroid and the thymus confused. In lab 2, you will learn a lot more about particularly the thymus. And I feel like I should just give you a heads up about lab 2 right now. In lab 2, we actually are going to be taking mice and identifying many of these immune organs. The mice will, the day before, say, be alive mice. And then right before lab, I will magically turn them into not alive mice. If sort of working with, and basically we're going to identify some of the organs that we're talking about here. We're going to take them out and we're going to make cell preparations that we're going to use for our cells for much of the rest of the semester. But if that's something that you're not comfortable with, we can talk. But one of the organs that we're going to be looking at is the thymus. So you'll know about the thymus much more after that lab. The second group of organs that are really important here are a group of organs called the secondary lymphoid organs. The secondary lymphoid organs are in this image of from your textbook in yellow. And you can see that there are a lot of them all over the body, labeled in a lot of different places. And so you can see that we've got things like the lymph node, we've got the tonsils, we've got the adenoids, we can think about the appendix, we think about the spleen, there's some specific intestinal organs. So there's a lot of different secondary lymphoid organs. I'm going to give you the punchline of kind of a sentence definition for a secondary lymphoid organ now, just to make sure you have it. But then I'm going to sort of back up for a little bit and try to tell you the bigger story of it to help it make more sense. So a secondary lymphoid organ is officially defined as a place where an immune response is initiated. So that a secondary lymphoid organ is a place where an immune cell first encounters its microorganism. And to be honest, we're usually talking about lymphocytes when we talk about that. Sometimes I think about a secondary lymphoid organ as where the party happens. And so immunologists spend a lot of time thinking about secondary lymphoid organs. I'll show you a lymph node in the mouse. You'll be getting the spleen in the mouse. But to really fully understand the secondary lymphoid organs, I have to tell you a little bit about sort of the immune system's relation to the circulatory system. And I'm probably going to run out of time while doing this, so I'm going to probably leave you on a cliffhanger. That'll be great. So this image is in some ways an immunologist's view of the circulatory system. The only thing that's missing here is that usually if I would draw a little heart over here, or you can imagine it like a really big pump that pumps liquid. And so you can think about a big pump that pumps liquid and goes through a hose. Like you can have a fire hydrant with a fire hose or garden hose in your backyard. You turn the spigot on full blast, right? And then we have all these little hoses that come off of it and get smaller and smaller and smaller until we get to capillary. It has this very tiny diameter, basically like one cell goes through it. Okay, so we have big pump pumping liquid through these hoses that are getting progressively smaller and smaller. So I want you to imagine your garden hose. Someday I'm going to do a demo in class of this, but I always forget to pull myself together. Imagine I have some big water pump, and I'm going to have the water go really hard from my water pump through my garden hose. What happens if you take that garden hose and you squeeze it and decrease the diameter that the water can go through? Yeah, Brianne. So what's going to build up? The water, and specifically is going to be a pressure buildup. So you're going to get like a big bubble. It might explode. What were you going to say, David? High water pressure, you're basically going to get explosion, right? So I just showed you how the circulatory system works. You guys all get that, like, you might get like a backup, the water's going to stop flowing, you're going to get an explosion, all these terrible destructions. Now I want you to look around. None of you have exploded while we were sitting here. You have all managed to have this happen without that explosion, without that massive pressure problem. The reason why you're able to do that is that all of the arteries through here are actually have selectively permeable membranes that let out water, that let out liquid. So what happens is the water actually gets forced out to balance out the pressure and keep you from exploding. And so I always learned originally, like, capillaries are so tiny, it's just one cell going through. Yeah, it's one cell going through because all the water got forced out. There's like no water. So you can see this is good. You are not exploding. No one has exploded during our class period. You are basically forcing all the water out of the arteries. Officially, this is sometimes referred to as the interstitial fluid. However, there's another problem that comes up because of this. This is where I'm going to leave you on a cliffhanger. If it just worked like what I just said, there wasn't an additional piece to all of this, that was what we're going to on the next slide, you would all be Jabba the Hutt. Because you would have all of this water that was leaving your blood and leaving your blood vessels, and then you would just be a big blob of water. So somehow that water has to get recollected. So you don't just become a humongous blob. And on Friday, we'll start with what happens there and how that relates to some other organs of the immune system. and then we'll move into the humoral innate immune response.